minutes for the last people to join the meeting, then we'll get started. Okay.
On us for the
Okay, good afternoon, everyone. I think we can get going. The rest can join us when they join. So very welcome to this SAMAC R&D webinar, or we also often refer to it as a virtual research focus in a nutshell. So it's basically just some feedback on some of our research projects through an online platform, because it's very accessible to all our growers spread over the country. The topic for today's webinar is genomics and genetic fingerprinting of macadamias. And we have Professor Zander Myberg and Melissa Reynolds and Mary Ranketsi. They are all from the University of Pretoria online. I'm going to hand over to Zander shortly. Just some housekeeping rules. Please remember to mute yourself. Nadira will also mute everyone from her side during the presentations. After Zander's presentation, you can just unmute yourself if you have a question for Zander, and then just please remember to mute yourself again for Melissa's presentation. So that's it from me. Very welcome. I'm going to hand over to Zander now. Thank you, Zander.
Very good, thank you. Can I just get a sound check? Can everybody hear me fine?
Perfect, thanks, Zander.
Good, yeah, I'm joined here in my office by Mary Ranketsi. Mary is a PhD student working on macadamia genomics, and it's actually through Mary that I got involved in macadamia. Our work is in forestry, so we work with the forestry industry. So if I can go to the next slide. There we go. So we are the Forest Molecular Genetics Program. We've been doing this for about 20 years now, working with the main forestry company, Sappi and Mondi initially, so working on eucalyptus breeding, and more recently also on pine breeding with York timbers and Safcol, and then also some other species the small growers are growing wattle, the acacia species. So we have lots of experience in developing genomic resources and then using that information to build tools that can be used for molecular breeding. So the macadamia industry in that sense is in a good place because most of the technologies that we have can be applied. Oops, there's somebody else competing with me. Somebody needs to be muted. Good, thank you. On the national stage, there's a wider set of organizations that are involved with us. So there's the core consortium, part of the Forest Molecular Genetics Consortium. But we also work with our forestry, South Africa, our industry association. And then, as I mentioned, with our work with Macadamia, we have now started working with SAMAC. And we also closely associate, in fact, most of the work initially that we've done in collaboration with the Macadamia Protection Program. So Dr. Gerda Fourie here at FABI, so they are a sister program in FABI with us. Then we work widely internationally with lots of international institutes. Currently, we have a very large project ongoing, sequencing over 3,000 eucalyptus tree genomes. This is with the Joint Genome Institute in Berkeley, in California, and also the Hudson Alpha Institute in Alabama. So these are all institutes that are, at this point, what is happening on an international scene in terms of molecular breeding is that we have gone beyond sequencing the initial reference genomes and meaning getting the first blueprint for any particular species. We did that for eucalyptus already in 2014. Now we're in a process where we're doing germplasm sequencing, where we're getting the genomes of all of the breeding parents, for instance, in a breeding program. And so we're moving into a new phase of molecular breeding that will be genome informed. As I mentioned, we've been doing this for 21 years already. It's a very successful partnership between industry and government and academia. We have funding from the main departments, Department of Science and Innovation and Technology Innovation Agency in South Africa, and then working with lots of different breeders in the companies that are helping us to apply the technology. So on our side, we're working on the lab side of the technology, but a big part of the molecular breeding is actually applying and working out how to use that information in practical tree improvement. And it goes well beyond just improvement. It also is talking about deployment and propagation. So we're looking at nurseries, nursery practice, making sure that plants are accurately propagated. That's where we found many, many problems. You can imagine if you've done 20 years of breeding and then you go and propagate the wrong clone, you've lost all of that genetic gain. And unfortunately, there are horror stories like that in industry. So macadamia genomics, I can say, is in the post-genomic era, just like eucalyptus. Post-genomics meaning that most of the initial reference genomes have been done, and we are beginning to move into a new era where we will do many different genomes. We will also be looking at different macadamia species. So there's about 3 or 4 species that we suspect have contributed the gene pool that is currently being grown in South Africa. And so this means that the time is ripe to now translate this information into practical tools. And those tools can be anywhere from maintaining our germplasm, designing orchards, to actual genomic selection and breeding, having a predictive model that can tell us at seedling stage what the performance of the plant will be. So I'm going to hand over quickly here to Mary and tell you and let her tell you what has been done in the international arena and what we have done, and then I'll get back to you again.
Thank you, Prof. So hello, everyone. So just briefly with Macadamia Genomics. So it has the typical plant-sized genome that we know of. So nothing unusual with macadamia in terms of its genomics. It has hello? Has 14 chromosomes and a relatively medium genome size ranging between 750 and 850 megabase pairs. This is depending on the cultivar or the species that you're sequencing, and also the very typical number of genes that you find in most plant species. So with macadamia, it ranges anywhere between 30,000 to 38,000 genes. And as we know, that most commercial cultivars are a hybrid cross between the two species, which is the integrifolia or the tetraphylla, as well as some potential three-way crosses might be in the mix. So far on the international front, in terms of the sequencing that has been done, we have the current genome, the 741, which is now acting as our current reference for macadamia. In addition to other integrifolia genomes also sequenced was the 344, which I think most people know, as well as the Chinese version, which is the GR1 cultivar. Then with tetraphylla, there was also sequenced recently, and another study that's been going on with the Australian researchers. They've managed to sequence now all 4 macadamia species. That includes the Jansseni as well as the ternifolia. Okay, so coming closer back home to South Africa, us adding genomic resources, we've managed to sequence 3 high-quality genomes for macadamia. So the first one is the Santa Ana, which we think is a macadamia tetraphylla from California. However, it is possible that it may be a hybrid. We just need to do some further work to confirm that. We also have the 695, which I'm sure everybody knows and loves. So this is a hybrid between the integrifolia as well as the tetraphylla. And then finally, the 791, which is a special kid on the block, which may be a potential tri-hybrid cross with an additional ternifolia in its genome. So in terms of the statistics that we get, it's more or less in range of what we see with the other papers that have been published internationally. And we have a high completeness score ranging between 98 and 99% of the genome that has been sequenced or contained within our assemblies. And again, the number of genes are within the range of what we see internationally. So now that we have some potential, of the blueprint of macadamia, how then can genomics assist macadamia growers? So firstly, we published this paper in 2022, whereby we used microsatellite markers, which Melissa will talk more about, just to get an idea of the genetic relationships of the macadamia that is present within South Africa. So you can read more about that. And this also enabled us to be able to differentiate and fingerprint different cultivars. So what that then allows us to do is efficient management of the resources and propagation that we have, which is essential when it comes downstream to breeding processes. In addition to this, it can also help with the optimal design in establishing orchards, because we know that macadamia is an outcrossing species and cross-pollination does assist with a higher nut set. And then finally, further on down the line is now developing this molecular genome-assisted breeding tools that Prof. Zander just mentioned. All of this can eventually then help us to develop DNA marker systems that are linked to traits of interest that breeders are interested in, such as the tree growth, flowering time, because we know there are some special cases like the 695 and 791, as well as increasing yield and oil production, which is also really important. And then finally, an integrated management for disease resistance of our macadamia orchards. So all of this together, we've sequenced the genomes, we've got the blueprint. What can we do then from there on? So we've taken the example of the macadamia nut oil biosynthesis. So this is the oil that is produced by macadamia nuts. As we know, macadamia has more than 75% of total fatty acids, which is the oils, making it the highest of one of the highest of the tree nut species. What makes macadamia even more special is that it is the only botanical, ready-to-eat source of palmitoleic acid, which is an omega-7 fatty acid. And there's a lot of interest now in palmitoleic acid because of its good for your health. So it's been shown to improve your cardiovascular health and such. Also, palmitoleic acid makes up between 7% to 36% of the total oils. So this was found over a range of studies. This includes the different macadamia species. And previous studies have found that there are two specific genes that may be directly associated with the conversion of the production of palmitoleic acid, and that is the SAD6 and the FAD5, which I've just abbreviated here for us. And just an example of what we've done within our study. So we've looked at 4 different gene families, which include the SAD, as well as the FAD gene families. But here we just have an example of the SAD gene family, whereby we're showing that, so we've got here the macadamia cultivars are in the blue font on the left-hand side of the image, and then on the right-hand side of the phylogenetic tree, we have other tree species, all in the green font. So this is just comparing the gene orthologs of the macadamia oil-associated genes to that of other tree nut species, as well as other oil-producing species such as sunflower and olive, for example. And what we see is that within macadamia specifically, there is an expansion of these genes, particularly of the SAD6 gene. So far what we're seeing is this could be corroborating what we are seeing in literature, but obviously we need to do more work towards that end. So I will end it there and pass over back to Prof. Thank you.
Okay, very good. So yeah, that was just a quick snapshot on how we are using these genomic resources. So now those high quality genomes that Mary has sequenced become a discovery platform. So it gives us leads into genes that can then be used for molecular breeding. And if we want to start breeding for different oil content, but the same can be done, of course, also for fruit development and flowering, many other traits. So I quickly just switch topics and mention that the genome also gives us the information to work with to identify the trees, of course. So out of the genome sequence, we were able to find DNA markers that can be used as a fingerprint. I'm not going to talk too much about that because Melissa Reynolds, who is the manager of our platform that is doing the DNA fingerprinting, is going to give you a detailed analysis of that. But just to mention that we have these 13 DNA markers that are spread out all over the genome, and we are using it. We had an honor student this year that started doing some of the pedigree confirmation and cultivar analysis, so we can fingerprint these cultivars. And Melissa will tell you a little bit about the initial results. We already know that there are potentially, in some cases, some misidentification that has happened historically already that needs to be replaced. But I won't say too much more about that. Melissa will talk much more about that. The plan is that there will be a SAMAC. There is a SAMAC project. It's funded by SAMAC, and there will be a central database that can be used. Because if you want to do this work, you need the references. So somebody needs to establish what is the 695 DNA fingerprint, and that you do by doing it at least from 3 different places and establishing a consensus fingerprint for that genotype. And then in the future, other people can map against that. So as I said, Melissa will talk about this right after, because we now do have a Macadamia DNA fingerprinting service that is accessible to growers. I want to talk about another aspect which will be empowered with these genome-wide DNA markers that we're developing, and that is this concept of ancestry mapping. There's some similarities between macadamia and eucalyptus in the sense that much of the work we do in eucalyptus is actually done on F1 hybrids these days. So we are developing hybrid varieties, and we think that most of the macadamias have some hybrid origin, although none of them have, as far as I know, been completely analyzed and completely confirmed what that genomic composition actually is. And here's what we've done for eucalyptus. So let me show you an example here. So Eucalyptus grandis is the one main species that is bred in South Africa. And then it's combined with, where's my mouse now, Mary? Maybe I should turn on my, I'm going to turn on my laser pen here. It's easier to see. That's it. So if you have reference sets, so here I'm just showing the first 5 chromosomes, or is it 5 or 6 chromosomes of 11 of eucalyptus. So we had a reference set of maybe 50 or 60 pure Eucalyptus grandis trees. So these are wild, unimproved trees. And same for Eucalyptus nitens. So Eucalyptus nitens is a more cold-tolerant eucalypt, and they make the F1 hybrids so that they can plant these in higher, so it's got the good growth of grandis, and it's got the cold tolerance of nitens, and that means you can start growing it up the escarpment and go into areas that are more cold prone. Now, what is important about this is if you have a genome-wide set of markers like we had here for about 20,000 markers throughout the whole genome, we can, for every position in the genome, basically paint the genomes. It's like chromosome painting. So when it is yellow, it means that it's carrying only Eucalyptus grandis DNA. And if it is blue, it's only carrying Eucalyptus nitens DNA. If it is gray, it is carrying exactly 1 copy of Eucalyptus grandis and 1 copy of nitens. So an F1 hybrid looks like this. Here's the genomic profile of a particular individual. Is 1 tree, is an F1 hybrid, the 11 chromosomes, and we can paint the entire chromosome. And in this case, the software identifies that it is a perfect F1 hybrid. What does it look like if you go to an F2? So here's a case where an F1 individual, a GN hybrid, was backcrossed to one of the 2 species. In this case, it was to grandis. So immediately you see more of the DNA going back to having 2 copies of the grandis DNA. So both the paternal and maternal copy of the chromosome comes from Eucalyptus grandis. But there's still some component of the genome that remains having 1 copy of grandis, 1 copy of nitens. And then we can discriminate that then in turn from if an F1 self, for instance, or an F1 by F1, so an intercross of a GN by GN hybrid, then you have this kind of profile. So some parts of the genome carries 2 copies, both the paternal and maternal copy is nitens. Some parts of the genome is grandis, and other parts of the genome is still an F1 hybrid. Why is this important? I think this is going to be one of the things that we will have to do for macadamia, is to sort out what are the actual genomic compositions, because it turns out that one of these components, let's say in the end of chromosome 3 here, might be carrying the cold tolerance that is required. So if you're trying to breed trees for a particular environment, or it might be the disease resistance that we require in a certain area, so we can begin to associate the genetic makeup with the traits of the tree. And this can be done across the population. Here's a case study where we then work with, I think this was with Mondi, and we worked across their grandis and nitens parental populations, identified cases where there were some cases here where they thought these were pure Eucalyptus nitens trees, but it turns out that these 2 are actually F1 hybrids. When we screened the F1 hybrids, we identified that there's 1 individual in there which is not an F1 hybrid. Actually, it fits with this group. It looks more like a backcross individual, so it's an F2 hybrid. And inside there, we found again other cases of this actually is not a backcross but an intercross. So it allows us to do genetic resource management and sort out the relationships of the trees and understand what it is exactly that we are working with. And I think this is one of the initiatives that we would want to take in the next couple of years. So what it will require, and we are talking to collaborators in Australia, is having a reference set of 50 or 60 trees or more that represent each of the wild species in Australia. And we know already at University of Queensland, some work has been done to sequence the genomes of the wild unimproved species. Then lastly, I want to just quickly talk about genomic breeding. So how will we go about to do genomic breeding in macadamia. So one of the things to realize is that most of the traits that we will be working on, if you think about yield, is going to be polygenic, meaning there could be many genes involved. And to do that, it means to be able to track that diversity throughout the genome, we will probably need tens of thousands of DNA markers. Currently, with the set that we use for the fingerprinting, is only 13 markers, so maybe one marker per chromosome. Here, we're talking about hundreds or possibly thousands of markers on each of the chromosomes to be able to track all of those variants. And there are ways to do it now that we can identify that by genome sequencing. So we can sequence a diverse set of macadamia genotypes, and that would be called a reference panel. So this is something we're now also going to be doing for eucalyptus breeding, is establish a reference panel. So basically all of the parents that potentially could be used for breeding in the future, you want to include, because then all the progeny DNA would be represented in there. Then you can identify the DNA markers in those reference genomes in the parents and the gene variants that are in those parents, and associate those with traits of interest, whether it's flowering, nut yield, nut quality, whatever traits can be measured in those trees. To do that, some hard work is going to have to be done. Breeders and growers will have to come together, and we will have to develop a training population or multiple training populations. Typically, we're talking about several hundred to at least 1,000 trees. It's somewhat unusual in horticulture to have so many. So I'm talking from a forestry perspective, where people already are talking about using 10,000 or 20,000 trees. But in horticulture, what we can see typically, it's done with a few hundred trees. I'm making a case that we should be making a training population of at least 1,000 trees that can then be analyzed with these genome-wide markers. And then once you have that, you can then build a genomic selection model that can be used to predict the breeding value of plants. So that, in that case, if you were to measure, for instance, nut yield on those trees and build a model for that trait, you can predict at seedling stage already what that's going to be. So you don't have to wait the 5 to 8 years to see the full phenotype. You can make much faster progress. In that way, you can save 5 to 8 years on your breeding cycle. And in a way to think about that, people always ask, is it worth the cost? It's going to cost maybe, let's say $50 per tree. And the question, one of my good friends in Brazil, who's also in this business, always will ask, what is it worth to have, let's say, 5% genetic gain 8 years earlier than expected? It is worth a huge amount of money. So it will definitely pay for itself to win this. And the other thing is, we will have to speed up the breeding to cope with climate change in the future. So this is one of the ways in which we are going to do this also in forestry. So how does it practically look? If you look at the genome, so as I said, Mary has already sequenced the genome. So this is a very small part of the macadamia genome. It's just hypothetical. I've drawn it myself. And so we've got all of the genes that she has identified. We don't know which of them are particularly involved in a trait, but for any particular trait, there will be a number of genes, hundreds to thousands of genes in some cases, that are involved. And the task we have is to find DNA markers that track these traits. So if you have markers that spread out over the genome and those that are closer to the genes that are involved, you can use statistics essentially to find that association, find the marker linked, the trait linked markers, and those markers together then become predictive of the trait. If you aggregate all of that information together, each of these might be explaining a small proportion, less than 1% of the genetic value or merit of the plant. But as you also sequence the entire genomes and find all the different variants that are present across all of your cultivars, the accuracy becomes increasingly high, because you're tracking all the diversity that's in your breeding population. So that is where we are heading. We've already put forward a proposal to SAMAC to start sequencing a reference panel for molecular breeding. And so just to summarize then, what we've done here at University of Pretoria is we have the first 3 reference genomes for 695, 791, and Santa Ana. The DNA fingerprinting service is ready. We've already started doing some projects and receiving samples now on a more routine basis. We have started a genetic linkage mapping project, and this is looking at the cross of 695 by 791. We predict that there's going to be all kinds of interesting traits segregating in that cross. So we're working with Duncan McGregor of iBreed on that cross, and that is a SAMAC-funded project. We're planning to, in the next 3 to 5 years, get ancestry mapping as an application for macadamia. I think that's going to be important to really resolve the hybrid nature and understand the adaptive value of these cultivars. We're planning to sequence a panel of at least 24 genotypes of relevance for breeding. It might not mean every single cultivar we have. It will only be those cultivars or varieties that will be used as parents in breeding programs, and then offer genome-wide genotyping service for macadamia. Ultimately, what we're trying to do is to develop a genome-assisted breeding platform for macadamia, and also training young people like Mary here with me as the future molecular breeders that can serve the industry. So with that, I will just refer you to our website. So if you go to just Google fmg.science, you will find the Forest Molecular Genetics website. And on there, we've got a website on Macadamia Genomics, and we'll be releasing information through that website. So, yeah, and thanks. So you can see we've already worked with this Wynand and Duncan, looking at their trees. We're genotyping a lot of trees already. And so we're getting our teeth into, and I've fallen in love with macadamia molecular genetics. I quite like it. So I look forward to interacting with more people in the industry about molecular genetics of macadamia. Thank you. Good.
Thank you,
Any
Zander.
questions?
That was great. Yeah, any questions for Zander?
Stop sharing maybe, so I can see who is asking a question. No questions?
No questions. It doesn't look like it. I think we can move on to Melissa. And then after Melissa's talk, if somebody would like to address a question to you after her talk, we can do it in that way as well. So thank you, Zander. Next up is Melissa Reynolds.
Thank you, Alria. I just want to double check that you can all see my presentation.
Yes, we can. Thanks, Melissa.
Okay, perfect. I assume you can hear me if you've answered. Okay, so thank you to Zander and Mary for doing a very nice introduction to macadamia. I will present a little bit on our project to establish a reference database for macadamia. But before that, I would just like to introduce the precision tree breeding platform. So the precision tree breeding platform is embedded within the Forest Molecular Genetics program. And our aim is to make DNA marker technologies accessible to predominantly the South African forestry industry, but also other local tree crop industries, academic researchers, and looking specifically at this point at small-scale farmers and production nurseries. So Zander has mentioned the value of focusing on production nurseries just to minimize human error. And this is something that we think is going to be quite applicable within macadamia. So if we take a look at the platform itself, we have a number of different people. But before that, I'll just give you a background on who we are and how long we've been operational. So we've been operational since 2008. And in that time, we have fingerprinted almost, well, I think it's probably just over 100,000 trees, predominantly forestry species. We focus on two different types of DNA markers, microsatellites, which Zander's mentioned, and SNPs. For macadamia, we're just focusing on microsatellites. We operate with several different genera, mostly eucalyptus, a bit of corymbia, pine, acacia, and more recently macadamia. And our main applications are identity verification, parentage analysis, and looking at species composition, and then the application of DNA markers to molecular breeding. We also have a development component to the team. I head that up, and that basically allows us to take the research that students produce, such as Mary, and apply it into an actual breeding tool for industry. So our team consists of 6 people at this point, permanently part of the team, and then we have interns as well. We have the DNA team who prepare samples and extract DNA. We have an analysis team who do the fragment analysis using DNA markers and analyze the data. And then the leadership team, which consists of me and Dr. Zelda du Toit-Boshoff. So she is the operational manager of the team. So most people would first contact me if they want to use any aspect of our services. And then I hand them over once we've onboarded them to Zelda, who handles projects coming in and reporting and communicating the progress of projects to users of the platform. So we have a pipeline that begins with sample preparation. We receive mostly leaf samples, which we dry and we grind in this Geno grinder, which allows us to process 576 samples in 2 minutes. So it's quite a high throughput platform. Once we have dried leaf tissue that's ground up into a fine powder, we can then transfer that to our Octopure DNA extraction robot. Is that a question or somebody joining the meeting?
It was just someone joining at this
Okay,
very late
no
stage.
problem. Okay. Okay, so we have our Octopure DNA extraction robot. This enables us to process 768 samples within 2 hours. We also have reusable plasticware, which reduces the cost. And overall, this is a very cost-effective method of preparing our DNA. In total, we process around 12,000 samples a year. But we have the capacity to drastically increase that because of this robot and our Geno grinder. So we definitely have capacity to expand into different industries at this point. Then once we have the DNA, we do our DNA fingerprinting. For this, we can comfortably process around 384 samples a day because we're relying on the sequencing facility at the University of Pretoria. Zander mentioned already that we have 13 microsatellite markers that we use for analysis in Macadamia. And these came out of Mary's PhD project and also published sources. Once we have our DNA marker data, we then perform data analysis. And in the case of Macadamia, that would predominantly be cultivar identification and then pedigree analysis. So this is our pipeline. Once we do all of our analysis in-house, we then provide a written report to the user of the platform. And if needed, we also have a meeting where we can present those results, discuss them, and consider follow-up questions as well. So this pipeline is very well established. We've been doing this for many years, and yeah, it runs pretty smoothly. So now that you understand what we do in terms of what service we can offer, I just want to look at the basics of what exactly is a DNA fingerprint, just to make sure that everybody is on the same page. So if we look at DNA markers, a DNA marker is a specific position on the chromosome or throughout the genome where we observe genetic variation. So this genetic variation is represented by peaks, and then these peaks represent alleles. Well, we call those alleles. So alleles represent the genetic variation for an individual at a specific DNA marker. If we look at how these are inherited, if we have a seed parent with a specific allele, a pollen parent with a different allele at the same marker, the seedling would inherit a combination of alleles from the seed parent and the pollen parent. So alleles are inherited from the parents, one from each parent at each DNA marker. So if we combine all of the alleles at the different DNA markers throughout the genome, in the case of Macadamia, we're looking at 13 different DNA markers, we end up with a combination of alleles at all of these markers, which is the unique DNA fingerprint for that individual, or in this case, that cultivar. So if we have a cultivar with our DNA fingerprint, we have an unknown individual, we can compare that to the reference fingerprint for that cultivar, and we can use this to identify matching individuals and assign cultivar identity to an unknown individual. We can also compare DNA fingerprints between cultivars and use that to differentiate between different individuals or different cultivars. So these are the ways that DNA markers can be applied in terms of cultivar identification. Let's now look at how DNA fingerprinting can add value to breeding and farming in macadamia. So if we look at cultivar analysis, just to recap, each tree genotype, in this case we're talking about a cultivar, has a unique DNA fingerprint. So this cultivar identity can be mixed up in field or in the nursery in many different ways. One example could be mislabeling in the nursery. When cultivars or material are shared between different farmers, they could be renamed, and we have definitely seen instances where that could have happened. There could also be mutations, and we could also have situations where siblings could be accidentally referred to as a single cultivar, which would result in some genetic variation within that cultivar. So DNA fingerprinting can be used to maintain cultivar integrity by identifying situations of mislabeling or where siblings are perhaps referred to as a single cultivar. So in order to make use of DNA fingerprinting in cultivar identification, we need to have a reference fingerprint for each of the cultivars of interest. So once we have a reference fingerprint for those cultivars, we can take a tree with unknown identity, we can fingerprint that, we can compare it to all the fingerprints in our reference database, and then assign cultivar identity to the unknown, based on what we already have in our reference database. So it's really important to have reliable reference DNA fingerprints to be able to compare the fingerprints of unknown trees in order to establish the identity of that tree. So you would have seen Mary referred to her paper that was published in 2022. And at this point, the samples that Mary fingerprinted and presented in that paper, you would have seen this in her slides earlier, those up until now represented, well, each cultivar was represented in that set by a single sample, and this has been our DNA reference database for these macadamia cultivars. So as Anda also mentioned, the importance of having reference DNA fingerprints that are confirmed by multiple different sources. And that is really important. So at this point, this reference database has shortcomings in that it's only represented by a single sample. So when we talk about a DNA fingerprint that is confirmed in multiple different sources, we talk about a consensus reference DNA fingerprint. So for example, if we were to have cultivar 1 planted out in an orchard, cultivar 2 and cultivar 3, if we were to sample a single tree from an orchard representing cultivar 1 and use that to establish a DNA fingerprint, if that tree happens to be something else and not cultivar 1, we could have an incorrect reference fingerprint for cultivar 1. So what we try to do is we try to sample several individuals from an orchard, or preferably from different sources. We aim for 3. If we can establish that their DNA fingerprints are the same, then we can establish a consensus cultivar fingerprint for that specific cultivar. And we can do this over many different cultivars and many different sources and establish a reference database from that. So that is something that we are now doing in collaboration with SAMAC. We have an honor student, James Smith, Zander mentioned him just now. So he has been working on establishing a cultivar reference set for key cultivars in South Africa. And this is funded by SAMAC. So what we did, Alria and other members of SAMAC sampled in Limpopo, and we went on a field trip into Mpumalanga and sampled at a few different sources. And the results that I'm going to show you now are showing the comparison of samples from Limpopo, from Mpumalanga, and from our FMG diversity set, which is that which was developed by Mary, represented by single samples for each cultivar. Okay, so we begin with 695, which is Beaumont, the most widely planted cultivar in South Africa. Fortunately, we were able to confirm that all the samples sampled in Mpumalanga matched each other and the reference that we have, and all of those in Limpopo matched the reference. So it was a nice, easy result. We were able to establish a consensus reference DNA fingerprint for Beaumont. So that's all nice and tidy. If we look at a different cultivar, 788, we sampled in Mpumalanga and Limpopo. We had samples from Mpumalanga that matched each other, but didn't match our referenced fingerprint for 788. And these matched 508. So these were from one farm in Mpumalanga. Then if we look at the samples that we collected from a different farm in Mpumalanga, again, those didn't match the reference fingerprint that we have in our database for 788, and we reassigned these to 741, so they appear to match our reference fingerprint for 741. Then we had some samples that were collected in Limpopo, also supposed to represent 788, and again, they didn't match the reference that we have on file, and didn't match anything else in our database. So we clearly have a unique genotype in Limpopo and two different genotypes being cultivated as 788 in Mpumalanga. So what this means is we cannot establish a consensus fingerprint for 788. And based on that, we will need to go and sample further in different areas. We have actually collected samples, which we can now genotype, additional ones. We just started with a subset of 3 per farm. So we will need to do quite a bit more fingerprinting for 788 to resolve the true identity of that cultivar. Then if we look at A4, we have, is that a question?
No, Melissa, you can continue.
Okay, thank you. If we look at A4, we have samples from Mpumalanga and Limpopo that match our reference in the FMG diversity set. We also have some samples from Mpumalanga that don't match that, and we're reassigned to 695. These are all from one farm. And then we have samples from Mpumalanga, well, a single sample from Mpumalanga that also doesn't match that reference, but did match our reference for 772. So here again, we have several samples that do match and probably represent the true reference fingerprint for A4, but it would be wise just to fingerprint a few additional samples to make sure that that is actually the true reference fingerprint. Then if we look at NALMAC, so we have NAL2, 2 samples from Limpopo did not match NALMAC 2, but were reassigned to NALMAC 26. This is not unexpected based on some discussions we've had with industry. Then if we look at NALMAC 26, we have 1 sample that does match our reference and 1 that doesn't, which was reassigned to NALMAC 2. So based on discussions, we already know that there could be some confusion between these 2 cultivars, or perhaps mistaken propagation from specific nurseries. So this will be really important just to fingerprint a little further and to confirm the true identity of these cultivars. So again, here we need to do further confirmation in order to establish the true identity of each of these cultivars. So this is just giving you a little bit of an overview of the work that James has done for his honours project, just to establish true identity of important cultivars within South Africa. Our FMG diversity set also consists of many more, it's over 100 samples from different cultivars maybe that are not widely propagated within South Africa. So we do have good representation of many different cultivars. So the value of this when it comes to farmers is really just to be able to confirm the cultivar identity at the point of sale. So this can be when validating a preferred cultivar for a specific end product. It can also be for ensuring that the correct cultivar is received from a supplier. And I think this is probably what's most important to the farmers. We have a situation now, we've had several projects coming in from different people, and one of them is to try to understand what happened because some cultivars that were planted as one cultivar is not flowering as expected and likely to be a different cultivar. And this has implications for propagation on that farm or productivity on that farm. It's also important for nurseries to fingerprint what they are cultivating and propagating within their nursery so that they can improve the buyer's confidence when purchasing specific material for a specific site. So it would be really helpful for nurseries to genotype and to be able to sell material to farmers with a DNA fingerprinting result attached to that. And this obviously helps farmers to avoid costly mistakes when planting incorrect cultivars and only finding out several years later when they're not flowering as expected. So this is basically the value of DNA fingerprinting in cultivar identity. So I've just focused on that. We have done projects on parentage analysis. Our panels are working very nicely for that. So Zander touched a little bit on the work that we're doing with Duncan McGregor on that. So just to wrap up, I just want to talk about how you go about accessing the service. So we have a few steps that have to be followed. Firstly, it's important to register as a user. We have a registration form that can be accessed via our website. And this will just allow us to understand where you're coming from, get you onboarded as a user. We like to connect with people online or via a phone call before any projects are planned or anything comes in. And this is just to make sure that we have the resources available to answer the questions, that we plan the project correctly, that we make sure references are sampled if we don't have them or something like that. And just to make sure that we're all on the same page in terms of how that project will run and the results that the user is expecting. Once we have discussed the project and where you want to go with that, we require an agreement just to be signed. And this is just acknowledging the terms and conditions of our platform and the prices that we charge for each of the different analyses. Once those things are complete, a user can make a booking. Again, we have an online booking form that can be accessed via our website. And this is to book the project. So we have, as I mentioned, a very high throughput platform. We process around 1,000 samples every month. It's really important that we have a booking in place and that everybody knows when to submit samples and that we are sure we can assist you with that at that specific time. We generally approve. We don't normally have any issues. And then you can also use that booking form to request a sampling kit. So we are busy just in the final stages of printing nice boxes and stuff. So we can send you a sampling kit with all the necessary consumables in there to go out and sample. We have many people being a little bit unsure about how to go about this. So we are more than happy to have an online meeting where we can explain how to go about sampling. We also have in-field sampling guides, And within a week or so, if you go to our website, you'll be able to see little videos on how to sample, how to set up your plates, how to use these sampling kits. So we have all the resources in place to do that. And then once you have received your sampling kit, you can go ahead and do your leaf sampling. These are the components you will most likely receive in your sampling kit. These little biopsy punches you can use to sample leaf discs and pop those directly into a plate. Those can then be shipped to us. And the details, as I mentioned, for doing this is all in our field sampling guide. Okay, so that is basically how to use the platform, what we do, and the value that it will bring to farmers. Any other information that you want, you can access on our website. As I mentioned, we're going to be adding quite a bit in the next week or two. So there will be a lot of resources there just to understand what we do and how it works and how you can use the platform. And obviously you can contact me as well at any time if you'd like to discuss fingerprinting or how to go about sampling or anything like that. Okay, thank you. Are there any questions?
Yes, I have a question, if you don't mind.
Yes.
Your cultivar references that you compare the samples from, where did you acquire that?
The cultivar references. Okay, so initially Mary's reference set came from, I think it was, Mary, you can correct me if I'm wrong, but from the ARC. And then from there, we worked with Alria to identify some farms in Mpumalanga, which we went and sampled at. And then Alria and Lisa from SAMAC also went and sampled in Limpopo. I think that was at Lavubu, and based on what I see in the sample sheet, possibly at a few other farms in Limpopo as well. Alria can clarify if needed. So we are aiming for sampling from different sources. And the reason for this is to make sure that we are not basing our reference on an incorrect propagation at one specific farm. We are comparing from multiple different sources. And that's really important, specifically to hopefully represent different nurseries as well, that we don't want to just sample from many sources that have all got it from the same nursery. So that is important. Yeah.
Thank you.
Maybe I can just also jump in there. Peter, is it you that asked the question?
Sorry,
Anyway.
it was Cornell.
Cornell. I think the majority of the initial samples that Mary used came through the ARC because that's the path that most of the cultivars that came into South Africa followed historically. Then the Mpumalanga samples, we sampled from orchards that we identified with some of our R&D committee members. Especially Alwyn de Pre and Stefan. And then Lisa, who is part of the SAMAC team, and Elsha Uber sampled in Lavubu and in Sanin. So that is where all the different samples came from.
Thank you.
William, you have a question?
Thank you, Alria.
William, you are muted if you are speaking. I think while we wait for William, if there may be any other questions, I'm just trying to see.
In the chat box, there is a question. Hi, I would like to know if some of the leaves sampled for A4 in Mpumalanga, but match 694 on your database. Did these trees portray A4 phenotype as well?
So that would be very difficult, I think, for Melissa and Zander to answer because they just get a small piece of leaf. What we are now planning on phasing in with the sampling sheet is to record some basic information on how the tree looked, so leaf shape and that type of thing, growth form, to at least point us in the right direction so that we can fall back on some of those phenotypic characters. But we also GPS all samples that are collected, so we can now go back to those trees and assess those phenotypic characteristics.
Yeah, thanks, Alvia. I think just to add to that, once we have completed, well, we have done the analysis, but once we've put this into a nice little report for everybody, we will send those reports to the specific farmers so they can see the outcome of this. They were very kind to allow us to sample as potential references on their farms. So it will be helpful for them to be able to go back and to look at what didn't match where it should have, and to actually go and look at those trees and to give us some feedback. So we are definitely not the experts on any of the cultivars. We will need feedback from the farmers to just tell us whether those results make sense and how they would apply it and how they would understand it, and potentially where any mismatches or mix-ups could have occurred. So there will definitely be feedback to the farmers and discussions and follow-up on that to resolve that kind of issue. But yeah, it's a very good point to raise. One does actually need to take these results and to go and have a look in the back at the farms and have a look at those trees and make sure that it makes sense. And that's, yeah, that they can trace back potentially where that problem came from.
Any other questions?
Just
Prof.
a
Maya,
comment. Can you still hear me?
Yes, you
Yes,
can.
I can.
Sorry, we have temporary load shedding here. Just one thing that is going to be very important for the industry is, as we uncover these cases of misidentification, it's going to be really important that you correct the labeling. I think some of the mistakes we've made before with the forestry industry is that we realized that after maybe a decade of having done the fingerprinting, that nobody actually went back and corrected that the identity of a tree sitting in an orchard, or if it's a hedge in a propagation facility. So it's really important to, if we want to weed out all of these cases, to either correct the identity, or in some cases with the forestry, they have pulled out entire hedges and just destroyed the plants and started over in some cases where there are mix-ups. So it's maybe a discussion that had to be held. Otherwise, these mistakes get propagated, and before you know it, you've done all the work, but you're still sitting with all of the error.
Yeah, thanks. And I actually just want to follow up from what Zander said there. So that's a very good point that Zander's raised. Many times new users of the platform don't necessarily expect specific results. So sometimes the results are a bit of a surprise, things don't match as you expect or whatever. And to be able to apply the results that we provide to you, it's so important that you make sure you track the trees that you sampled. So we provide on our website, you can download a sample collection sheet template, which just gives you an idea of what kind of information you should record when sampling. And we also require the sample data and any metadata associated with that to be added to a sample submission sheet. And in there, as Aria mentioned, the GPS coordinates of each tree should be recorded. Also the position in the orchard, if you want to do that as well. But it's so important to record what tree you sampled from and to make sure that you associate that with the specific tube or well in the plate or something that you are submitting to us. So if anything is a surprise, you can go back to that exact tree to confirm the phenotype compared to the results that we give you, or to look back at your records and see where it came from or something. So the value of fingerprinting is mostly in how you go back and apply it. And if you don't record where it came from, you can't apply it. You can't relabel a tree. You can't maybe cull the correct tree in the orchard or something like that. So if there's anything that I would like people to take home from this, please record which tree you sample if you send anything to us. It's so important in terms of applying our results. Yeah. Okay, are there any other questions?
Yeah, Alria, just a general question, and this is maybe for people that travels a lot and some expert on the farms. 849 is an important cultivar to us, and I'm just familiar myself with the cultivars because I'm new in this. Which area is known for 849s? Because I don't see it. I'm from Zululand. I don't see much here except on the farm that I'm working on. And South Coast, I don't see a lot. And Levubu, not a lot. So other areas, which area is known for 849s where we can get a good 849 reference, which is important to us?
Peter, thank you for putting me on the spot. I am unfortunately not a technical advisor, so I have no idea, but I know a lot of very good technical advisors, so I can ask them. But sorry, I unfortunately really don't know. I know Alsha is online. Do you perhaps know Alsha? Sorry for putting you on the spot, Alsha, sorry.
No, it's fine. It's fine. We have more 849s in Mpumalanga than in Limpopo. And I know that we did sample 849 here in Limpopo, and it was the same then because Melissa didn't say that there was a discrepancy, right? So yes, I would say we need to use the Mpumalanga samples as baseline, but we feel quite safe saying that 849 is 849, right?
Sorry, just to interject there, I just pulled up a few examples. So, yeah, maybe let's just look at the results before we confirm that 849 there is correct. Okay. I'll just have to go have a look. I didn't add everything. We had many different cultivars represented in that set. So these were just a few examples showing different scenarios.
Yeah, it's already scary to see the variations that you've got just with these scenarios.
Yes, definitely.
Okay, thank you, Alsha.
Okay, I think there's no further questions. I think if anyone has any questions that you think of afterwards, you are very welcome to contact myself or Zander and Melissa. I think thank you very much, Zander and Melissa and Mary, for your time. We really appreciate it. I think you presented some very complex concepts in a more simple and very visual manner. So thank you very much. We really appreciate it.
Alria, can I come in quickly with another question, a last one?
Okay, Peter.
Thank you, Alria. Well, I have all the people listening, 788, because I see you used 788 there, and you have a good fingerprint for that. Is it widely known that we have a Natal 788 and then call it the inland or a northern or Limpopo 788? Is that because I see the 788s on Levubu that I can see, okay, this is the 788, and coming to our place, it doesn't look the same. In South Coast, different story. Is that why? Is that known?
Sorry, yeah, go ahead, Aria.
Maybe I can start and Melissa, you can finish. So I think a lot of people have that feeling. What we haven't done, Peter, is to sample KZN. So our strategy was to start with Mpumalanga and Limpopo and see how much variation do we see, and then we'll move on to sampling in KZN as well.
Okay, thanks.
The idea is that most of the trees, or not most, but a lot of the trees in KZN would have come through Mpumalanga or Limpopo. So that was our strategy.
Yeah. Okay.
Can I just ask a question on that? Is that the one where, so this is new to us. When we went to one of the farms, they said there's a light variety and a dark leaf variety.
Yes.
Is that what we're talking about here? Okay,
Yeah.
so we did sample different representatives from 2 different farms, and they are definitely different, the light and the dark varieties from those 2 farms. So I do think that there's a merit in your question, and we definitely need to resolve that further.
Okay.
Yep.
Thanks.
Yeah, so when, maybe Peter, just background, when Alvain prioritized the orchards where we sampled from in Mpumalanga, he explicitly added those 2. I think the question now is which one is the real 788? So is it the dark green one or is it the light green one? But yes. Okay, thank you very much, everyone. I think we've gone over time. Thank you very much. Thanks, Mary and Zander and Melissa. We really appreciate it. We will share the recording of this webinar on WhatsApp and on our YouTube channel, and we will also load it onto our website. So thank you very much, everyone. Have a good afternoon.
Thank you, everybody.
Pleasure.
Thank
Bye-bye.
you.
Bye, everyone.